All right, so today we're diving into a world that I find just fascinating.
Oh, me too.
It's like we have this superpower that lets us see what's happening
Yeah,
inside individual cells.
absolutely.
We're talking about intracellular flow cytometry,
Mm-hmm.
or ICFC for short.
ICFC, yeah.
And it's really like unlocking these secrets that are hidden inside every single tiny cell.
It really is. And the things you can analyze with it.
Oh, tell me.
You can look at proteins and signaling pathways and even gene expression
Wow.
all at a single cell level.
It's pretty amazing.
It's like having a tiny detective
Right.
working on the case for us.
Microscopic detective.
Exactly.
Yeah, so for this deep dive, our guide is an article from Assay Genie.
Okay.
It's called Navigating the Intricacies of Intracellular Flow Cytometry. Key Considerations.
Makes sense.
And believe me, there are a lot of details that matter, especially if you're aiming for accurate research results.
Oh, absolutely.
Right.
It's a really powerful technique. But it's not as simple as just looking at a cell under a microscope.
No, not at all.
There are a lot of steps involved
Right.
in preparing the cells and making sure that the data we collect is reliable.
Okay, so imagine this. You're trying to study a specific protein
Okay.
deep inside a cell.
Okay.
The first challenge is, how do you even get access to that protein?
Right.
You can't just crack the cell open like an egg.
Exactly. They're very delicate.
Right.
And that's where this idea of permeabilization comes in.
Permeabilization.
We need to make the cell membrane permeable to antibodies
Okay.
so that they can get inside the cell.
So the antibodies are like our little detective.
Yes.
But they need a way to get
Yes,
in.
they need a way in.
Right, exactly.
Right. Okay, so how do we do that?
So there are a couple of methods that the article talks about.
Yeah.
The first one
Yeah.
uses this gentle agent called Saponin,
Saponin. Okay.
and it basically creates these tiny pores in the cell membrane.
Uh-huh.
So almost like poking tiny holes.
Just enough for the antibodies to slip through.
So it's like we're carefully opening a tiny door
Exactly.
just to let those antibodies in.
Just enough. And what's great is it's reversible.
Oh, that's good.
So the cell can actually repair those pores
Wow.
after the antibodies have done their work.
That's really important.
Yeah, especially if you're working with delicate cells,
Right.
like immune cells, where you really need to preserve
Right.
their natural structure.
That makes a lot of sense.
Mm-hmm.
But I imagine there are some cases where a gentler approach like Saponin isn't enough.
Yes.
What are the alternatives?
So if you need something a little stronger,
Okay.
there's the option of using methanol or formaldehyde,
Okay.
and these fix and permeabilize the cell
Got it.
by cross-linking the proteins.
Oh.
It's kind of like taking a snapshot of the cell.
Oh, okay.
The structure is preserved,
Yeah.
but you might lose some of the finer details.
Oh, so it's kind of a trade-off then.
Yeah,
You get stronger permeabilization, but you might lose some information.
exactly. Along the way.
That's why choosing the right method is so important.
Yeah.
It really depends on what you're trying to study.
Right.
Are you looking at things that disappear quickly?
Right.
Or are you looking at stable structures?
Gotcha.
So it really depends on your research.
Okay. So now we've gotten our antibodies inside the cell.
Okay.
How do we make sure they bind to the right target?
Ah, that's a great question.
Right. Because cells are packed with all sorts of molecules.
Yes, absolutely.
We don't want them just grabbing onto anything.
We don't want them going rogue.
Right. Exactly.
So that's where antibody specificity comes in.
Antibody specificity.
Yes.
We have to make sure our antibodies are like highly trained bloodhounds. They need to find our specific protein and nothing else.
Otherwise, we're going to end up with false positives,
Exactly.
like a detective arresting the wrong person.
Exactly.
So how do we prevent that?
So we have this process called titration.
Titration, okay.
And it's basically adjusting the concentration of the antibodies
Okay,
until we find that sweet spot
got it.
where they bind strongly to their target,
Yeah.
but don't create a lot of background noise.
So it's like fine-tuning the signal,
It was
making sure it's
just-
nice and clear.
Exactly.
Okay, now I remember reading about these fluorescent tags
Yes.
that are attached to the antibodies in ICFC.
Yes.
What do those do?
Ah, the fluorescent tags.
Yeah.
These are really cool. They're
Yeah.
like tiny beacons
Okay.
that allow us to actually see what's happening
Yeah.
under the microscope.
So they light up.
They light up. When
Right.
you hit them with a laser,
Uh-huh.
they emit light at specific wavelengths.
Okay.
And then we can use that light to identify and quantify
Wow.
our target proteins.
So we're basically lighting up the cell from the inside
Yes.
to see where our protein is hiding.
You got it.
That's very cool. But it sounds like choosing the right fluorochrome
Yes.
is pretty important.
It is. We need to make sure it works with our equipment.
Right.
And it doesn't overlap too much
Right.
with other fluorochromes that we might be using.
Because we might have multiple antibodies in there.
Exactly.
Each with a different tag.
Uh-huh.
And we don't want those colors to get all mixed up.
We don't want it to be a big mess.
Right. It would be like a painting where all the colors have run together.
Exactly.
How do we prevent that?
So that brings us to compensation.
Compensation.
Yes, and that's a mathematical correction that we
Right.
use to subtract any overlapping fluorescence.
So we're cleaning up the data,
We're cleaning it up.
making sure we can see each signal clearly.
Yeah, it's like adjusting the color balance on
Mhm.
your TV to
Yeah.
get the most accurate picture.
I like that analogy.
Yeah.
And luckily, modern flow cytometry software
Yeah.
has made this a lot easier,
It has. We
right?
can automate a lot of that process.
That's great. But even so,
Yes.
understanding the principles
Yes.
behind compensation is still really important.
It is, absolutely.
So researchers know they're using the right settings
Right,
for
exactly.
their experiment.
Yeah.
Okay, so that's how we get antibodies into cells,
Yeah.
make sure they're binding to the right targets,
Mm-hmm.
and even how we deal with
Yeah.
potentially overlapping signals.
Mm-hmm.
But I know there's more to this story.
Oh, yeah, we're just getting started.
Right.
We've only just scratched the surface.
Stay tuned,
Stay with us.
because we'll be right back to talk about some of the really cool applications.
Yeah, the advanced stuff
Yeah.
of ICSC.
All right, we'll be right back.
We'll be right back.
Okay, so we're back.
Back again.
And we've covered a lot of ground so far.
We have.
We talked about how to get antibodies into those cells
Yeah.
without, destroying them.
Right.
How to make sure those antibodies are binding to the right target.
It's all about specificity.
Right.
And even how to untangle those fluorescent signals.
Right, using compensate.
Exactly.
So I feel like we're well on our way to becoming microscopic detectives.
We are, but
Yeah.
there's still more to the story.
There's always more.
Always more.
So the article also talks about some of the advanced applications of ICFC,
Yes, some really cool stuff.
like intracellular cytokine staining
Mm-hmm.
and phosphoprotein analysis.
Yeah.
Those sound pretty intense.
Powerful techniques.
Okay, so let's break them down a bit.
Okay.
Let's start with cytokines.
Okay.
So cytokines are like the messengers of the cell, right?
Exactly.
They help cells communicate.
Yeah, they're essential for so many processes.
Right, like immune responses, inflammation.
Cell growth, development.
It's all about communication.
Absolutely.
But studying them inside the cell
Yes.
sounds tricky.
It can be challenging.
Why is that?
Well, because they're often expressed very transiently.
Transiently?
It means they kind of appear and disappear quickly.
Oh, so like trying to catch a fleeting thought.
Exact.
Okay, so they're fast. And on top of that,
Mm-hmm.
they're really small
Tiny.
and they could easily leak out of the cell.
Especially during permeabilization.
Right, when we're poking those little holes.
Exactly.
So it's not as simple as just adding antibodies and seeing what happens.
No, it requires some specialized techniques
Okay,
to make sure we preserve those cytokines.
so they don't escape.
Exactly.
Got it.
Yeah.
Now, what about phosphoprotein analysis?
Yes.
What makes that one so challenging?
So imagine the cell is like a busy city,
Okay, I like this analogy.
and the proteins are all the workers.
Doing their jobs.
Right, carrying out various tasks.
Okay,
Now, phosphorylation is like attaching a special badge to a worker.
a badge.
Yeah, it can give them new instructions
Okay.
or change their behavior.
So by studying phosphoproteins,
Yes,
we can understand how cells respond to different signals
exactly.
and change their activities.
It's like watching how the city adapts
Oh,
to
wow.
changing conditions.
But those phosphate groups, they're kind of like those cytokines, right?
They can be easily removed.
So we need to be extra careful
We do.
to preserve those modifications during the experiment.
Absolutely.
Okay, so we need special antibodies
Yeah.
that only recognize the phosphorylated protein.
Yes. And techniques that prevent dephosphorylation.
Got it. So it sounds like ICFC researchers need to be masters of timing and precision,
They do.
but it's worth it
Oh, absolutely.
because we're talking about understanding the language of cells,
Exactly.
how they communicate,
How they adapt.
and carry out all these processes that keep us alive.
It's fascinating.
It really is.
It's like eavesdropping on their conversations.
And then using that knowledge to maybe develop targeted therapies.
Exactly.
That's amazing.
It is.
It makes you realize how much we still don't know
We've only just begun to scratch the surface.
about this microscopic world inside us.
It's a whole universe
It really is.
in each cell.
So this whole idea of eavesdropping on cells and using that knowledge to develop smarter therapies, it's mind-blowing.
It is. It has so much potential.
Now that we can analyze all of this in such detail,
Yeah.
what's next for ICFC?
That's the big question.
What breakthroughs could it lead to?
The possibilities are endless.
I mean, it's amazing to think we can analyze this cellular communication in such detail already.
It is.
So what's next?
Well,
What kind of breakthroughs could ICFC lead to in the future?
that's the question that keeps researchers up at night.
Yeah.
I mean, it's a field just full of potential.
Yeah.
Imagine being able to pinpoint
Okay.
the exact cellular pathway
Yeah.
that goes wrong in a disease,
Right.
like cancer
Uh-huh.
or autoimmune disorders.
So it's like finding the faulty wiring in a circuit
Exactly.
and then, where to go to fix it.
And once you know where the problem is,
Right.
you can develop much more targeted therapies
Right.
that address the root cause.
And that could minimize side effects and make the treatments more effective.
Exactly.
And I imagine with ICFC, we could even track how individual cells respond to different treatments.
Absolutely.
Like in real time.
In real time.
It's like personalized medicine.
That's the goal.
Tailored to each patient.
Yeah, based on their own unique cellular makeup.
That's amazing.
And don't forget about immunotherapy.
Oh, wow.
ICFC is really helping scientists understand the immune system and how we can use it to fight diseases like cancer.
This whole idea of eavesdropping on cells and then using that knowledge to develop these therapies is incredible.
It's mind-blowing.
Makes you realize just how much we still have to learn about this microscopic world.
We're just at the beginning.
Yeah.
There's so much more to discover.
So if someone's listening and they're fascinated by this world of intracellular flow cytometry, where can they go to learn more?
Well, the Assay Genie website is a great resource. Okay.
They have protocols, articles, even tips for optimizing experiments.
That's fantastic. A one-stop shop for ICFC.
Pretty much.
Well, this has been a really eye-opening deep dive for me.
It is a fascinating topic.
I'm walking away with a whole new appreciation for the complexity and the beauty of the cellular world.
And remember, there's always more to explore.
Always.
Keep asking those questions.
Yes.
Stay curious, and who knows? You might be the one making the next big discovery.
That's a great point. It's a reminder that science is always moving forward.
Always evolving.
And there's always something new to learn.
Absolutely.
Well, thank you so much for joining me on this journey.
It's been a pleasure.
Into the world of intracellular flow cytometry. It's been fascinating.
It has.
Until next time, keep diving deep.
Keep exploring